My name is Monica Russa Gertz, and I'm graduating this semester with a PhD in neuroscience. I'm also the first winner of the NSF Graduate Research Fellowship in Mason's history. My research here at Mason was an interdisciplinary effort guided by a wonderful group of investigators from different fields. My dissertation was an experimental and computational investigation of metabolic demands on neuronal responses to focused ultrasound stimulation. To give you a little background, the effect of focused ultrasound is similar to shining a light through a magnifying glass. It can affect tissue at the focal point of the sound waves, but leaves all of the surrounding tissue intact. This platform is already in use clinically to treat Parkinson's and epilepsy by ablating damaged tissue. But it's also been found that using this technique at lower intensities can stimulate neuronal activity without damaging tissue, and has great potential for non-invasive deep brain stimulation. However, the mechanisms are not fully understood. Moreover, no one has investigated metabolic demands of focused ultrasound stimulation, which is important because even subtle changes in neuronal metabolism can have a profound effect on neuronal activity. Although my research was in neuroscience, my project included mechanical engineering and design to integrate ultrasound with microscopy so that I could perform my experiments. My experimental methods measured metabolic activity in the rat hippocampus by recording simultaneous oxygen and potassium responses to both electrical and acoustic stimulation. I combined this experimental data with a novel computational model developed jointly with Dr. Cressman and was able to tease apart the underlying mechanisms, as well as replicate our experimental results. Our results revealed different metabolic demands between electrical stimulation and acoustic stimulation. We also modulated neuronal responses to electrical stimulation using focused ultrasound based on model predictions. This implies that the model can be used to guide basic and clinical protocols to maximize effectiveness while avoiding damage. Mason also helped me with additional opportunities through its seminars. One such seminar led to an invitation to attend the NASA HRP conference. There I met Dr. Christopher Mason, who was a leading investigator in the NASA twins study, where twin astronauts, Mark and Scott Kelly, were studied before, during, and after Scott's 340-day mission on the ISS. This study resulted in the famous summary paper in Science. After that conference, Dr. Mason gave me an internship where I analyzed the biochemical signature recorded on landing for Scott Kelly. My first authored work was published in Cell Reports, discussing the biochemical role of in-flight muscle atrophy and regeneration on landing, and was also mentioned in CNN. My next step is as a postdoctoral associate in the Department of Genetic Medicine at Weill Cornell Medical College, where I will be applying my knowledge of focused ultrasound to develop localized gene therapy treatments for patients, among other projects. Many thanks to the faculty and staff of George Mason University for these great opportunities.